Hello everyone and welcome to another episode of Noir Yudarris. This is your host Mahmoud Noir. Today we'll be speaking briefly about lipids in biochemistry context. What are lipids made of? What are their structures? Type of lipids found around us. And stick around, like, subscribe, share the video, and don't forget to leave any comments or concerns or any future topics you want me to cover. Alright, shall we start? So lipids are amphipathetic molecules, meaning they exhibit both hydrophilic and hydrophobic parts. They have hydrophilic polar heads and they have hydrophobic tails. So their structure is basically this. This is hydrophobic tail. That's where lipids are. So phospholipids are made of a phosphate and an alcohol as a polar head joined to the hydrophobic fatty acid, and they are both joined together by a phosphodiester linkage, like glycerol for instance. Right? Glycerol can be a good phosphodiester linkage. And hydrocarbon chains, like this one, have two determining properties. They're either saturated fatty acid tails or unsaturated fatty acid tails. So saturated fatty acid tails, they have single bonds, no pi bonds. Single bonds, like I mentioned before, like sp3, they're weak and they are stable, and they have van der Waals forces, like for instance milk, coconut, palm oil, um, and then you have unsaturated fatty acids, and these include one or more double bonds, right? So here you see how they're single bonded, but with the unsaturated fatty acid they include double bonds and they have kinks. What I mean by kink, or what is meant by kink, is that a double bond that causes a bend in the structure, and it makes it difficult to stuck and solidify. For example, of an unsaturated fatty acid is an olive oil. And then you have phospholipids with unsaturated fatty acids make up more fluid region of the phospholipid region. So phospholipids with unsaturated fatty acids, they make up more of the fluid region. Then you have glycerol phospholipids. Glycerol phospholipids are phospholipids that contain glycerol backbone, like we see here, linkage and linked to two fatty acids by phosphodiester linkage. Alright, but there's also an ester linkage here. So an ester linkage is all connected to CH3 and a CH group, so a C and a C group here. These are the ester linkages. And then you have the sphingolipids. Sphingolipids have sphingoid backbones. They're long, they're non-polar, and they have polar head. This is a sphingosine, or a sphingoid backbone, basically. It looks like this, with one double bond and then a lot of CH3. Ceramide are simplest sphingolipids. They're made with single hydrocarbon as a head group, and then you have sphingomyelins. You have an example of that as phosphocholine or phosphoethanolamine as polar head group. And then you have gangliosides. These are glycolipids that have polar heads composed of oligosaccharides with one or more a N-acetyluramic acid or NANA. They have no phosphate group, no glycosidic linkages. With these sphingolipids, I don't think it's really that significant in the MCAT, but also it's worth knowing as well. Next you have terpenes, which are lipids, and they are naturally occurring chemical compounds found in plants and some animals. These are responsible for the aromas and the flavors in like plants and some animals. So they are made of isoprenes, which are units, and what monoterpenes means is two isoprenes, sesquiterpenes is three isoprenes, diterpenes is four isoprenes. Terpenoids are derived from oxygenated terpenes, and they both feed into biosynthesis pathways that produce steroids. So terpenes are also significant for the production of steroids. Steroids are nonpolar molecules forming four-ring 4 rings of carbon, like we see here. You see, these are steroids, right? So steroid hormones, they act as hormones secreted by glands into the bloodstream, where they bind to receptors such as testosterone and estrogen and cortisol. So steroids are hormones, and they are responsible for binding and activating um, certain receptors. Cholesterol maintains the constant cell fluidity, and a low temperature keeps cell from becoming too permeable. Cholesterol maintains constant cell fluidity. Low temperature keeps cell from becoming too permeable. Prostaglandins produced from prostate gland and acts as paracrine or autocrine signaling molecules. So prostaglandins can act as signaling molecules. Next we have the vitamins. These vitamins are fat soluble. Okay, vitamin A, unsaturated hydrocarbon, used for vision. Vitamin D, liver, kidney, calcitriol, increase the calcium and phosphate intake. So vitamin D increase calcium and phosphate intake. Vitamin E characterizes a group of lipids called tocopherols and tocotrienols, which are antioxidants and prevent oxidative damage, which could lead to cancer. By the way, vitamins are really significant on the MCAT. They come up quite often, I would say. So try and really understand which are fat soluble and which are water soluble. Water soluble are like vitamin C and vitamin B and stuff like that. And then lastly have triacylglycerol. These are preferred energy long-term usage, which contain 1 to 3 ratio 1 glycerol to 3 fatty acids, and they're connected by ester bonds, and they are very hydrophobic. Usually lipids are most of them are hydrophobic, uh, and they have no charge except for just a couple which have a slightly polar side to them. I'm talking about the unit of the lipid as a whole, or molecule as a whole. And that's it. That's a wrap. Thank you so much for tuning in. Let me know if you have any questions, and have a blessed day.